Hi, and welcome to the beginning of section 3, the cell. All living things are made of cells, and you're going to see how the last two sections of study we did, starting off with macromolecules, then going into water and the cell membrane, are going to help aid our study on how we study the cell. Remember that there's four major macromolecules. We have carbohydrates, protein, lipids, and nucleic acids. And when it comes to water and the cell membrane, there's a beautiful relationship between the two. And we're going to talk about the cell membrane a little bit more, but also in relation to other structures within the cell. So here we go. Let's go ahead and start our study on the cell. And before we start going into the nitty-gritty on the inside of the cell, we're going to talk really quickly on how we study the cell. So the first part of our lecture is all very small. It's going to be about microscopes. So in typical outline fashion, we're in the cell. Roman numeral 1 is microscopes. So microscopes are essentially tools that allow you to magnify things, and you can actually get some pretty good clarity on what you're looking at. But there are two main types of microscopes I want us to just reference right now, because those are the most popular when it comes to cell biology studies. So the first one is the light microscope. What a light microscope is, it's a very simple microscope. This is one that you could buy for your home. You could easily buy them online. They're not super expensive. They can be if you want them to be. But what a light microscope does is that it shines a visible light, and that visible light is passed through the specimen you're trying to look at. That specimen could be a tiny little organism that's been mounted onto a microscope slide. It could be a drop of blood that you have on a microscope slide. These are usually things on a microscope slide. It's a thin piece of glass, and you put a cover slip on top of your specimen so you can focus your objectives that are attached to the microscope on that specimen. The objectives, you have many objectives within a microscope. Depending on the quality of your microscope, they could go anywhere from 4x magnification to 40x to 120x. It just depends on the style of microscope you end up buying. But what you need to know is that a light microscope uses visible light to pass light through the specimen, and that light gets bounced off. It goes through the glass lenses and it magnifies the image, which ultimately gets bounced off into your eyepieces so you can see the beauty and the detail of the specimen that you're looking at. So if you ever get your hands on the light microscope, I highly suggest you just do some fun outdoor play. If you have a pond nearby or some kind of body of water, you might be able to find some really cool life inside just one drop of water. It's pretty fascinating. So light microscope, visible light is passed through specimens. And magnified by lenses. And then ultimately that image gets put into your eyepiece so you can actually see what you're looking at there. And the benefit of, the benefit of a light microscope is that the specimen you're looking at can actually be alive. You don't have to kill the organism that you're looking at in order to visualize it. Some microscopes require that. We're going to get to that one in a second. But the benefit, this will be number 1. Number 1, the benefit, you're able to look at live specimens. The one drawback on the light microscope, although you get some really incredible detail on a light microscope, compared to the microscope we're about to study in a second, you get limited resolution and magnification with a light microscope. You can see a lot of good stuff. You could get down to the cellular level. However, you can't get into the ultra structure, that intricate detail inside of a cell. So it's limited in magnification and resolution. It's pretty good though. Now, don't get me wrong. I love my light microscopes. I see a lot of beautiful detail in there. However, in comparison to the next guy we're about to study, it leaves him in the water. So here we go. So that's a light microscope. Able to look at live specimens. They don't have to be alive, but they can be. And you're limited in magnification and resolution only to the strength of your microscope, really. So that's a light microscope. The other type of microscope that is excellent to use for cell biology, so you can see what the interesting detail is, the surface of the cell, the inside of the cell, is this one. It's called an electron microscope. An electron microscope is exactly that. It's a microscope that, instead of light, uses a beam of electrons to pass through or on top of the surface of your specimen. It depends on what you're doing. But it uses a beam of electrons to help visualize the specimen. So, uses a beam of electrons to visualize the specimen. And I'm simplifying this whole process a lot, but I'll talk about it in a little bit more detail right now. So what an electron microscope does, what you need to do to prep your sample that you're looking at in an electron microscope, you essentially, let's pretend we get a piece of skin tissue from an animal. And we want to look at the different cells within that skin, okay? So we get a piece of skin, and what we're going to do is we are going to prepare it in some kind of, it depends on the electron microscope techniques that are used in a specific lab, but there's many ways to prepare a specimen. Ultimately, what you end up doing is shaving little pieces of that tissue, so you get a very thin piece of tissue, kind of like a cross section. I want you to imagine if you have an ice cube, and you just take a razor and you just shave just a touch of one of the sides of the cube, and you get this very thin piece of ice, which in our world will melt immediately. However, if this is like a solid cube of wax, you could just easily just shave off this almost see-through piece of wax from that cube. That's exactly how you prep samples for electron microscopy. Now, what you do to that sample that you just did, this thin little shave, is that you stain it, what we say, with specific metals. And metals, when they get excited, their electrons will jump and they will emit certain light or fluoresce or do whatever the stain calls for it to do. And when you use this beam of electrons and you shine that beam of electrons on your specimen that has this staining on it, that allows specific structures in the tissue to glow, and you could actually see this amazing detail within the cell. Because the benefit to electron microscopy is the magnification. You can magnify to a point where you can see the individual phospholipids in a phospholipid bilayer. It's amazing. You can't get that level of detail in a light microscope. They're just not powerful enough. An electron microscope, thanks to the use of the electrons and the different staining, you're allowed to see that clarification, that resolution within an image. The only bummer about this is if you're going to do electron microscopy, you can't use a live sample. It's kind of obvious once you hear about how we prepare the samples, you can't keep something alive if you're going to do that. That's just not nice. So we don't do that. So the limitation are only what we call fixed. Fixed is another nice way of saying we had to actually kill the tissue or kill the sample in order to make this happen. You don't see this happening on whole body organisms. Like you would never do this to an animal. What you would normally do this to is a biopsy of tissue. So if I had a mole here I wanted to remove, I could get that biopsy taken out. And if I had an electron microscopist friend, they could prepare that sample of tissue and actually look at it. You would never do me and let that happen like that. So it's only for pieces of tissue. You don't have to worry about the whole organism. So only fixed tissue can be used. But the benefit is you get extreme resolution and magnification. Something like this we like to call ultrastructure. The benefit is you get ultrastructure, which is extreme magnification and resolution. So extreme magnification and resolution. That's the beauty of the electron microscope. There are two types of electron microscopes I would like you to know about. So number 3, you have 2 types. The first type is called a scanning electron microscope. Scanning electron microscope. The E with the negative sign, that's shorthand for electron in chemistry, so I'm just staying with my shorthand. So scanning electron microscopes. What they do is they literally scan the surface of your specimen. So this comes in super handy when we're talking about the microbial world. If you're thinking of a virus you're trying to study, one of the most important parts of a virus is learning about how the virus is encapsulated or coated, the viral coat. Because the coat is the first thing that interacts with us to infect us. If we can build up some kind of block against the coat, you can technically block the viral infection. That's the thought. So if you know what the shape and the structure and the details of that viral coat look like, you can start getting a little bit more idea on how it interacts with us, different drugs to fight it, stuff like that. So a scanning electron microscope will give you the ultrastructure of the surface of an organism. And on our website for here, I'm actually going to make a little slideshow for you so you can see the differences between light microscopes and electron microscopes. And I'll point out the difference between these 2 types of microscopes. So you'll know right away, oh, that was a scanning electron microscope, and the other one is what we're going to talk about in a second. So scanning electron microscope, this is obviously an electron microscope. It reveals the ultrastructure of the surface of a specimen. Reveals the ultrastructure. That's our structure of the surface. Of a specimen. And secondly, you can kind of guess where we're going here. I'm just going to erase this and put it up here because I'm running a little low. If one type of electron microscope looks at the surface, you can imagine there's a whole lot of information to be gained by looking at the inside, and that's exactly what this one does. Little b is called a transmission electron microscope. A transmission electron microscope allows you to reveal the ultrastructure of the inside of whatever you're looking at. The inside of a cell, the inside of a virus, the inside of a bacteria. These are super small things. You can't get that kind of resolution on a light microscope. You get ridiculous detail on an electron microscope. So a transmission electron microscope reveals the ultrastructure of the inside of the specimen. Okay. So keep a lookout for that on our website. You'll see that attached to this lecture. I'll have a PowerPoint slide, basically with really beautiful microscope pictures for you to compare. And you can see the beauty to be found in a light microscope. Compare that to the beauty you find in an electron microscope. You still see magnificent stuff, whatever you're looking at. And I know recently for my kids, I just bought, for my kids at home, I just bought handheld microscopes that you can buy and just go out in the backyard and magnify whatever it is you're looking at with using a very a small little handheld light microscope. Super easy to use. You literally just point and look. And even with that low grade of a microscope compared to what you would find in an academic lab, you still see some really neat detail, stuff you would have never known. Even looking at a piece of fabric, looking at the details of this fabric was pretty cool. So there's a lot of stuff out there to be seen. So that's it for microscopes. And now that we know how microscopes are studied, how cells are studied, let's talk about the cell. And I am going to actually save that for our next video because I want you guys to take a moment to go ahead and download that PowerPoint slide presentation and just check out the different images you can find using microscopes, depending if it's a light microscope or electron microscope. And I'll even probably put some pictures from a fluorescent microscope. That's another type of microscope that uses fluorescent dyes. If you shine it with a specific wavelength of light, it allows these dyes on the specimen to glow, and you can see the location of certain things that you're trying to locate in a cell. So the imagery we find in microscopy, that in itself is its own field. So if you're into this, you could always become a microscopist and just image things all day long. And it's wonderful. You actually provide a ton of information because how something looks helps reveal their function, structure, function. It all goes hand in hand. So the field of microscopy is really growing because there's so many new microscopes that are coming out now. And I'm just showing you the tip of the iceberg. It's huge. So come on back. We'll start getting into the nitty-gritty of cells, and first we're going to talk about the two different types of cells the big families of types of cells that you find out there, and then we're going to start moving inside the cell and talk about all the different lovely structures inside of a cell and how they function to keep the cell going. So come on back, I'll see you soon. Thanks.